Can you imagine how it would feel to be in this situation? Not being able to eat bread, apples, oranges, or hummus? Or are you even one of the 15% of people to whom this is reality? We are iGEM Vienna, and in 2021, we are tackling neglected gastrointestinal disorders. Irritable bowel syndrome, or IBS in short, is often overlooked. We have no molecular markers for diagnosis, and only recently, the cause for most IBS cases was confirmed: FODMAPs. FODMAPs is short for fermentable oligo, di, and mono-saccharides and polyols. They are a very diverse group of carbohydrates that can be found in most foods. Our guts themselves do not produce the necessary enzymes to break down FODMAPs, but some of the bacteria that are living in our guts do. Therefore, FODMAPs act as dietary fiber and prebiotics. Sounds like a good thing, right? Well, FODMAPs are osmotically active, which means that they draw water into the intestine. Additionally, when bacteria break them down, they are turned into gas. This is no problem for the healthy belly, but for some people, these processes irritate the gut, and they experience abdominal pain, bloating, diarrhea, and headaches. So far, we have no effective treatment but to avoid all foods that are high in FODMAPs, and patients often end up with long lists of foods that they are not allowed to eat. That is, if they even find professional help. Because in reality, this disorder is still not talked about enough. Many patients go on an odyssey of trying to find which foods they can or cannot eat. This not only puts them at risk of nutritional deficiencies. We talked to patients who told us that they also experience a lot of emotional stress, that they were uncomfortable to eat out, and they were generally at a loss of how and where to find help.
With our project Friendzyme, we tried to develop a solution that would allow people with FODMAP sensitivity to eat even high FODMAP foods. Because of the limited time, we decided to target one major subgroup, namely fructans. Fructans are among the most common FODMAPs, and they are responsible for problems in most FODMAP-sensitive patients. Our idea was to create something like an enzyme pill, but better. Fructans are fructose polymers, meaning they can be enzymatically degraded to fructose, which then can be further converted to glucose. However, since fructans are omnipresent, it would be quite inconvenient to take a pill every single time you want to eat something that contains them, like you would do for fructose or lactose intolerance. After all, there are not many dishes that do not contain garlic, onion, or grain. We spoke with several patients, and they all agreed that they would prefer to take medication less frequently. So, we needed a way to make these enzymes work in the body long-term without the need to swallow multiple pills a day. We found that the biofactory working directly in the gut would be the best way to achieve our goal. We ended up with a three-way approach. If you watched our promotion video, you will recognize following parts. We started with genetically modifying our lactobacillus to express endoinulase, levanase, and invertase, the weapons needed to break down fructose. The digestive tract is a harsh environment that can easily kill bacteria. In our promotion video, we used the submarine as a metaphor for our polymer scaffold, which would be used to encapsulate lactobacillus, protecting them from extreme pH and keeping them in place, while at the same time allowing the release of fructan-degrading enzymes. Under such circumstances, this capsule would leave the body after just a few hours. Therefore, we planned to modify our scaffold in a way that it would adhere to the mucosa of the gut.
We wanted to find the best expression cassette for our system. Therefore, we tried different secretion signals, promoters, terminators, and even various versions of our enzymes. In total, we added 15 basic parts to the registry. We decided to use Golden Gate cloning, also called Type 2S system in the registry, as our cloning method. Here, you start with simple DNA blocks of your sequences, such as promoters. In the first step, they are cloned into the first backbone called backbone 1. In the next step, the individual sequences are cloned into promoter, enzyme, terminator assemblies in backbone 2. In the last step, multiple such assemblies can be cloned into a single backbone, backbone 3. All those steps we performed in E.coli, as it is easier to handle, and only then switched into lactobacillus. To make this switch from E.coli to lactobacillus possible, we developed a novel backbone 3 for the Type 2S assembly system. One that works in lactobacillus as well as in E.coli. It contains an erythromycin's resistance and allows for an easy switch between the host organisms. Considering all our promoter, enzyme, terminator assemblies, we designed nearly 100 novel composite parts. Furthermore, to quantify the strength of our promoters and lactobacillus and to improve the documentation of existing ones, we compared them using mCherry as a signaling protein. In the end, we were able to show the expression of our proteins in lactobacillus, quantify their activity, and compare different expression cassettes. All those things do not come without struggle, failure, and setbacks. To help the future generations of iGEMers, we developed a detailed troubleshooting guide, which you can find on our wiki.
At the same time, we were working on our biocontainer. As a material, we chose cellulose sulfate and polydatmac, which are both biocompatible, and upon getting in contact, the negative charge of the sulfate and the positive charge of the polydatmac form a very stable polyelectrolyte complex, leading to the instant formation of a gel-like capsule. In addition to biocompatibility and stability, we wanted our scaffold to have another very important property, which is mucoadhesivity. This means that the scaffold will stick to the surface of the gut mucosa and remain there and release its enzymes over a longer time span. The way we decided to do this was via thiolation, meaning the addition of thiol groups, which then can form disulfide bridges with the cysteine residues in the gut mucosa, and since these interactions are covalent bonds, they are quite strong. Initially, we wanted to add the thiol groups directly to the cellulose sulfate. However, in the process, we unfortunately noticed the sulfate groups get cleaved off, so what we ended up doing was adding another thiolated polymer, chitosan, to the polydatmac solution. We were lucky enough to have one of the leading experts in thiolated polymers right here in Austria, Professor Bernd Schnöch from the University of Innsbruck, who helped us to conduct some experiments in his lab, some even involving real porcine mucosa, to show that our thiolated chitosan indeed has mucoadhesive properties. We also tried out different types of cellulose because the source of the raw material can make quite a difference when it comes to properties. Also, we tried out polydatmac of long and medium chain length. Then we went on to do our encapsulation assays. We were able to achieve very promising results when using cellulose sulfate and polydatmac together with live cells. And also when we added our thiolated chitosan, we were able to show that stable capsules are formed. However, then unfortunately, we ran out of time, so some more fine-tuning would be necessary regarding the ratios of the components, the chitosan, and the cell density in order to achieve optimal results.
In the creation of new therapeutics, the exact dosage is extremely important. Of course, clinical studies were off limits. This is why we modeled the degradation of the most common fructans, inulin and levain, to fructose and glucose. Our simulations should give us an estimate on how much enzyme is needed to degrade the fructans of one pizza margherita before they reach the large intestine, where they are known to trigger symptoms for patients suffering from IBS. Our results suggested that the fructans would not exceed the clinical threshold and would be degraded within 2 hours and 15 minutes, which is well below the average residence time of 3.5 hours in the small intestine.
From the beginning of the project, safety has always been our #1 priority. We understand that using genetically modified organisms in the human body is undeniably a difficult topic. This is why we developed a multi-level biosafety system that addresses the concerns of policymakers, specialists, and the general public. Our concept is designed to prevent the growth of GMOs in the body and in the environment. It starts with choosing a probiotic, generally regarded as safe organism for Friendzyme. We chose a Lactobacillus plantarum strain with natural auxotrophies. Additionally, our polymer capsule not only protects bacteria, but also acts as a biocontainer, which was shown by us to be effective. In the long run, we would like to stably integrate the genetic construct into the genome and to add a quorum-sensing-based kill switch that is activated when the bacteria get released from the capsule. We hope that this concept brings us one step closer to using the potential of GMOs as therapeutic delivery machines, and that one day Friendzyme can help people with all kinds of food sensitivities. But it doesn't have to end here. Mucous membranes are present throughout the body and make for an amazing target point for site-directed and extended-release drug delivery. We can even imagine a non-hormonal contraceptive, where the fructan-degrading enzymes are replaced with a spermicide. This way, Friendzyme could provide a basis for all kinds of applications in the medical field.
The average development time of a novel drug is 9 years. We had 3 months. Still, during iGEM 2021, we managed to improve the documentation of existing promoters, create an extension to the Type 2S assembly system, establish and develop 25 novel parts, and clone our enzymes into lactobacillus. Furthermore, we encapsulated bacteria for up to 24 hours and laid the foundation for making our scaffold mucoadhesive. Our project was significantly shaped by the close communication between experts, IBS patients, the public, and our team. Still, having IBS is often downplayed as being just a picky eater, but we hope that our efforts help giving the disorder the attention it needs and remove stigmata attached to it. We want to send out a huge thanks to all our partners and sponsors who supported our team and helped making Friendzyme possible.